Hello everyone. Today, I would like to talk about several brain imaging methods for detecting Alzheimer's disease. First, I will talk about what is Alzheimer's disease and why people get it. And then, several bioimaging methods for diagnosis. Finally, we will look at the future. So what is Alzheimer's disease? It is a kind of dementia. There are 50 million people worldwide which is diagnosed for dementia. And 60 to 70% of them are related to Alzheimer's disease. The Alzheimer's patients experience cognitive defects such as loss of memories, loss of language skills, or disoriented. In this picture, we can see the healthy brain is much larger than Alzheimer's brain. The space between the brain tissue from an Alzheimer's person is also larger because of the brain atrophy. There are two main factors that cause Alzheimer's. The first one is called amyloid beta. At first, there is a kind of transmembrane protein in our cell called amyloid precursor. Amyloid precursor is cleaved by beta or gamma secretase, and the residue is called amyloid beta. In Alzheimer's patients, the amyloid beta will not be cleared by enzymes and form into amyloid fibril precipitation. They form into abnormal plaques between our neurons and lead to synapses damage. The second factor is called tau protein. Tau protein stabilizes the microtubules, which transports the molecular between our neuron cells. In Alzheimer's disease patient, the tau protein is defective and dissociated from the microtubules. So the transportation between cells can no longer be processed, and the tau protein form into neuron fibril tangles, which lead to cell death. The first method I would like to talk about is structural MRI. The full name of MRI is magnetic resonance imaging. The main concept of MRI is that the signal of radio frequency can alter the energy state of protons, whose angular momentum is polarized in magnetic field. And because the MRI signal is sensitive for different tissue characteristics, we can see volume changes with MRI signal. In this picture, we see 4 different stages of brain. At the last stage, from a patient of Alzheimer's, we see white parts in the brain, which indicates vascular damage. This is caused by white matter hyperintensity. And here is another example of structural MRI. This three picture is taken from the same individual once a year for 3 times. And the small h here indicates the hippocampal area. As the time went on, we see that the black space here between the brain tissue became larger and the hippocampus became smaller. This is the evidence of hippocampus atrophy. The second method I would like to talk about is functional MRI. The main concept is similar to structural MRI, but here we measure the signal from blood oxygen related index, such as blood volume or blood flow. In the picture, the blue part indicates inactivated region and the red part is activated region. Studies show that with aberrant functional MRI activity, the patient is also detected with amyloid beta signal. And we will talk about amyloid imaging later. The third method is called FDG PET imaging. FDG is an analog of glucose, which is the main energy resource of in our brain. And in this picture, we can see in the brain of a normal person, the FDG is activated, which shown in red. But in the brain of Alzheimer's patient, the FDG is inactivated. The last method is amyloid PET. As we mentioned before, amyloid beta is an important signal for Alzheimer's disease diagnosis. So here we measure the signal from amyloid PET. There are 3 different groups of patients in this picture. The first one is normal control. The second is mild cognitive impairment. And the last one is Alzheimer's patient. We see that in normal control, there are barely signal from amyloid deposition. With MCI, the patient already show activated signal from amyloid beta. And at the last stage, in Alzheimer's diagnosed patients, the signal for amyloid beta is the strongest. Here, we will look at a graph from several different biomarkers in different phases of dementia. We see that the fMRI test-related brain activation would decrease in the last stage of dementia. And the fMRI network connectivity and FDG PET metabolism decreased as the disease went on. The hippocampus volume, which is measured by the MRI, shows also decline as the disease is progressive. So now you know the basic of Alzheimer's disease and the diagnosis for it. Alzheimer's is a severe disease of which the scientists have already investigated for over a century. But until now, there are still no cure for it. The current treatment focuses on maintaining mental functions, managing behavior symptoms, and slowing down the symptom of the disease. So brain imaging is important for studies of new therapies, especially in the trials, to check the progress of the decline, to detect side effects, and so on. We hope that in the future, as we know more and more about Alzheimer, we can not only predict, but also effectively prevent it. Here are my references, and thanks for your listening.